Okay guys, so here we have our trays with the rubber end caps on them, and then we've got the combs that are in the trays, but there's no gel in there, and that's because I'm preparing it. Here we've got our solution. How do we prepare that? Well, this is a substance called TAE buffer. It's electrically conductive. That's going to help our negatively charged DNA move through the gel. I better turn this down. It's getting a little out of control. Alright, and then we have our agarose powder, which when I dump that in a second, I'm going to give it a nice stir, and I've weighed out a certain amount, and it's a polysaccharide, so it's going to help the substance actually solidify once it cools. And once this solution cools to 60 degrees, I can pour it into each of these trays. Then you'll notice that each of these little projections in the comb are going to make an imprint in the gel, and that's going to form the wells in which we will use micropipettes to dispense DNA. So your DNA is going to, well, it's being kept in the fridge as you can tell, but here we have some. So you're going to have several samples. Sample A, B, C, D, E, F. And you're going to take a sample, if I can get this open, here we go. Take a sample of DNA from each microtest tube, and you're going to put those into each well. So like A is going to go in the first well, and B is going to go in the second, and C in the third, and so on. Actually, you have 6 wells you're going to fill. There are going to be 2 at the end that you're not going to fill. And you want to make sure that you eject your tips, which there are no tips on here, but you know what they look like. You'll eject a tip in between each sample so there's no cross-contamination. And that's how you do it. So we have some fun to look forward to. So I've been stirring here for like maybe 10 minutes. 10 minutes. The key is to make sure it's clear. So it's at about boiling right now, 100 degrees Celsius, not quite. The powder was dumped in. If it's cloudy, you know it's not dissolved. It's looking pretty clear. But you might notice I have a few little clumps of agarose. It's not too bad. It's almost ready. This one over here looks pretty good too. You see a few flaky clumps, so I'm going to keep stirring this. This is almost ready as well, and I'll be pouring the gel soon. So I have finished stirring this, and the temperature is a little under 60 degrees Celsius. One thing you want to make sure, if you ever do this in college, is that you don't want bubbles. Bubbles are bad, they're evil. So if you pour gel, if you pour this to make a gel, and you have bubbles in your gel, then that can inhibit proper DNA migration. So I'm going to demonstrate how to pour these gels. So you'll notice that here are the gels. And I'm going to pour nice and slow because I don't want bubbles to form as I'm pouring the gel. So you'll notice that this one I started pouring and then I ran out of fluid in the other beaker, so I'm using this one now. Notice that I want the indents between the combs to essentially fill with this agarose fluid. Well, this will take about 20 minutes to solidify, not very long at all. I put a little too much in that one, but that's okay. It's not as good to demonstrate improper technique. Ooh, I poured that one a little fast. So you see a bubble. There's a bubble, one of those evil bubbles. See how that works? I got. It's all I've got. That's how you do it. These we won't even fill. Maybe tomorrow. Maybe tomorrow. One thing I forgot to mention is how critical it is to make sure that the setting on the fridge is not too cold. And I made the mistake one year of putting the gels in a fridge that was way too cold. I was using the faculty lunchroom fridge, and the problem is that the gels ended up fracturing because of the extreme cold. So keep it temperature wise, I'm not really sure what the exact number would be, but don't go crazy with the setting. Now let me explain the importance of the TAE buffer. Tomorrow morning when you guys do this lab, you're going to pour the TAE buffer that I prepare into your gel. Now just imagine for a minute, let's say that you've taken the rubber ends off of the gel, and you kind of have to pry it off with your finger. Actually, you're taking the rubber ends not off the gel but off the tray. And then imagine that you have a gel sitting in this tray, and you keep the gel in the tray, and you make sure that the wells, which let's say that the wells are on this side as opposed to being on that side. Let's say they're on this side. Well, notice that the electrode that's negative is on this side, and the electrode that's positive is on this side. You want the DNA to migrate away from the negative electrode to the positive end. So you make sure that when you put the tray in, you don't put it in backwards. You make sure that the rectangular wells are on the side closest to this electrode, because DNA has a phosphate backbone that's negatively charged, so it's going to naturally repel. In fact, actually, you put it in more gently. Let's say that there's a gel in this, and you just kind of gently make sure that it slides in between these notches, and then you just gently put it in, like that. And then you take the TAE buffer, and you're going to pour not directly onto the gel, but you'll pour into this space in here, and then it'll just fill. And then the idea is to make it fill until you know, it covers the gel. The idea is to make sure that electrically conductive fluid covers the surface of the gel. And then that's when you take your micropipettes and you insert the tip of your micropipette into the fluid. So let's say there's a fresh tip. I've punctured, using this tip, I punctured the parafilm that covers the DNA. Remember those little microtest tubes? You puncture the aluminum film, suck out the DNA, and then you transfer that DNA to, let's imagine that well A is here, and you transfer. Then you eject the tip, get another tip, puncture, the second micro test tube with another tip, and then you put that sample B into the second well. Just imagine there are wells right across. So that's how it's going to go tomorrow. And make sure, this is so important, that when you're loading DNA, you're not bumping the chamber, and you have to keep the chamber in place, because the thing is, if you pick up this chamber and you move it, the turbulence will cause, the turbulence of the TAE buffer will cause the DNA samples to unsettle and actually come out of the well, so you don't want to do that. And then let's say that you've successfully put your DNA in. The black is the negative. So basically you slide this on, just like this. I'm trying to do this one-handed so I can hold this camera, this phone. And you make sure that you gently slide it on evenly. And then these electrodes here, or these electrode wires, will be plugged in. So red on red, black on black, and then we choose our voltage. Of course, we plug the thing in. This is a power source. Undecided yet as whether we'll do 150 volts. I've found that it makes the gels really, really hot, and that can be an issue. Or we'll do 75 volts. It just takes longer, so I'll decide that on the fly tomorrow. All right, it's gonna be fun.